Also, I will be recording this session since a lot of people that couldn't attend asked us if we could provide it later. So good morning, students, faculty, and staff from the University of Texas at El Paso and any external audience that we might have. My name is Paulina Rios, and I am the president of the American Society for Biochemistry and Molecular Biology student chapter at Utep. As a scientific organization, we felt the necessity to raise awareness on the importance of the COVID-19 vaccine. Therefore, today we are co-hosting this event with the Utep College of Science. Dr. Kirkin, the Dean of the Utep College of Science, is here as well, and the School of Pharmacy. So our guest speaker, it is Dr. Jose Rivera. He is the Dean of the Utep School of Pharmacy, and he will be informing the community on the university's vaccination program. We are honored to have the participation of Dr. Rivera and Dr. Kirkin because we believe that especially Dr. Rivera's experience on the health field make him the perfect speaker to inform our community. Before starting this talk, I just want to make a brief announcement to the ASBMB members. Please make sure to stay until the end to discuss some important points. And for the rest of the audience, we will be taking a picture at the end. So if you can turn your camera on, that would be great. Without any further delay, please welcome Dr. Rivera. Dr. Rivera, the floor is all yours. And yes, we're ready to start. Thank you.
Okay, I was just unmuting myself. Okay, so one of the things that I want to do with this presentation is to really make it an interactive presentation. I don't want to be just giving a lecture. So if there is anything during my presentation that you want to ask, I'm okay with stopping and discussing issues. Now, the very first thing that I want to talk about is why are we doing this clinic? So I want you to reflect on yourself, but from my point of view, back in the fall of 2020, I started participating with a working group in El Paso, and it was called the Surge Working Group, and it's related to the as the cases for COVID were going up, some of the healthcare organizations and the Border RAC, which is a regional advisory council, came together to discuss how to address the increasing cases and hospital beds and ICU beds and the use of mechanical ventilation from this disease. And I started asking questions because I knew that the vaccine was coming, that it was expected to be available sometime by the end of the year. And then I was asked to join another group, which was specifically looking at vaccinations. This is from the Department of Public Health in El Paso. And of course, there were people from all over El Paso County. And as I started learning more and more, I realized that the capacity of the Department of Public Health was very limited in terms of being able to do a lot of vaccinations. And that's when I went to Utep administration, and I posted the question. I said, we should do something about this. Even if our contribution is not as big as some others, we should be able to do some of this ourselves. And I know that the School of Pharmacy was in a perfect position to try to help with this, because a lot of the elements related to this vaccine and the vaccine clinic we can address. So that's when a decision was made to go ahead and apply. It's a process that took quite a bit of work to get the application going. So this is an outline of some of the topics that I'm going to be discussing today. And for some of them, I do have slides, and other ones I'm just going to emphasize at certain points why that was important. But this is the outline of everything that I'm going to be covering today. As you can see, it's an extensive list. I want to just first introduce the issue of vaccines. They are not all equal. The history of vaccines comes from ancient Chinese literature as the first documented, and it was an attempt to try to do a vaccine for smallpox. But here in this table, what I want to emphasize is the different types of vaccines. And sometimes people hear vaccines and they think that they're all the same, and there's confusion about that. So I'm just listing in here the different types, the more common ones, and I'm putting an example and also a comment, because as you could see, live attenuated vaccines tend to have some of the highest responses for single dose, but they may have more side effects. And there is also, you have to be cautious about using it in patients that have immune deficiencies. But as he goes down, you could see the different types of vaccines and an example of what they are. And as you go down, you could see that booster doses are necessary, that you don't get as much of a response with a single dose. But ideally, especially with the messenger RNA, you have fewer side effects. Now, the application process was quite complicated, and it took quite a bit of gathering information from different resources in order to apply. The first application was to the Texas Human and Health Services. And in there, we have to document a lot of information in terms of who can do this. For example, qualification of the clinical personnel. We needed to assure that there was a sterile compounding expertise, vaccination training, that everybody needed to have a national provider identifier number, and it went on. The list was quite comprehensive. For this particular application, Chief Medical Officer was designated as it could be an MD, a PharmD, a nurse practitioner, and this is because of the PrEP Act. And the PrEP Act is basically the Public Readiness and Emergency Preparedness Act that allows for this to be the case. There was also a lot of information that we needed to gather related to the storage capacity for vaccine. So there were issues with having secure limited access to it, the types of freezers, using electronic digital temperature control, backup generators, and so on. So it was quite a comprehensive application. And it took all my days during the holiday break to work with others at Utep to be able to complete that application. At the same time, in order to finish the application with the state, we needed to apply for the MTRAC2, which is a Texas Immunization Registry. And it's done by electronic exchange of data. And for the purpose of the COVID-19 vaccine, we need to report to them within 24 hours of administration. And it's quite a tedious task. We have been able to go from individual data entry to more of exchange of large data through the FIX, which is part of the Paso Health Foundation. So now we batch the data and submit it to FIX, and they submit it to the state through this NTRAC system. So here is one of the issues that we have to deal with is the ordering, receiving, and storing of the vaccine. There are some issues related to the vaccine that we started receiving, and that's Pfizer. We decided to, well, actually the state decided to send us Pfizer because we have some of the capacity to store ultra cold vaccines. So the system that they use is called the Texas Vaccine Allocation and Ordering System. And in there, we have to go in, select the vaccine quantities, and what vaccination group are we targeting. When we receive this vaccine, there's a lot of issues related to the actual transportation of this vaccine. And in there, I have a picture of what the container looks like. It does come, the Pfizer vaccine does come in dry ice, And it does have digital temperature control devices that are wireless that tracks the temperature of that container throughout the process. Once we receive it, we have 3 minutes from the opening of the box to putting the vaccine in a -80 ultra cold freezer. This vaccine is very delicate, so we have to do it very quickly. So they give you 3 minutes for us to do that. And of course, there is a lot of issues related to the inventory control. Usually, we were receiving 975 doses in 195 vials. Now that's going to increase, but basically, inventory control is critical in transportation and documenting how it moves, not only when we receive it, but when we actually take it out for preparation. We have to document temperature control throughout the process. Okay, so this is the vaccine that we're using, is the Pfizer BioNTech COVID-19 vaccine. As you know, uses messenger RNA technology, and it received emergency approval use back in December 12 for individuals that are 16 years of age and older. Now, for the purpose of Utep decided to only vaccinate 18 and older. And this is because you need consent from parents, and we did not have that many individuals that, to our knowledge, that were between 16 and 18 years of age. Now, the clinical trials that were done were multinational, and in order to get that emergency approval use, there were a total of about 43,000, more than 43,000 people enrolled in a phase 3 global randomized placebo-controlled trial. And it was a 2 vaccination that were separated by 21 days versus placebo. So it was documented that it had an efficacy of about 95% in protecting, and this is after the first dose. But to interpret this, remember, you give the second dose 21 days later, so this is using the 2-dose system. Of course, the type of side effects were relatively mild, and pain at the injection site, anybody that receives an IM is going to have some type of pain or tenderness. But there were a few other issues. Fatigue and headaches were common. And the number of anaphylactic reactions was relatively low. The most recent data documents about 10 anaphylactic reactions, 10 to 11 per million people. Now, this is a severe reaction that can be fatal if you don't treat. But I'll show you a little bit of information in terms of what we're doing in the clinic to have the resources to manage this if it does happen. Of course, the storage was critical. The ultra cold, usually this will allow the vaccine to be viable for about 3 to 4 months. But once you move it around, it decreases the life of the vaccine. So it depends on what stage you are. And in there, it lists the different types of duration for that vaccine, depending on where they are. Now, a little bit about synthetic messenger RNA technology. Everything is based on DNA to protein and the gene expression process. So as you could see here, if you insert some messenger RNA, that goes directly to the ribosome, where then it produces the protein that we're looking for. Now, the COVID or vaccine is to target the SARS-CoV-2 gene expression and replication in host cells. The way this vaccine works is by developing a target spike protein that is on the surface of the corona virus. And as you may know, that the virus enters the cell and it goes through the process of the duplication and then is released back. And some of the most important side effects are associated with the effect on the lungs, causing inflammation. And of course, severe cases require mechanical ventilation. There are some drugs that have been approved for use to reduce the mortality, including as simple drugs like dexamethasone, which has been around for a long time. So in here explains a little bit more of how this works. So synthetic messenger RNA molecules are encoded to basically produce a response from the immune system to that spike protein on the surface of the virus. This is modified for stability and prevent them from being recognized by the immune system. So you do package it with liposomal membranes with some carbohydrate molecules. And this also produces stability and the ability to deliver to the target places, tissues and cells. So you do get that response of high levels of a spike protein. Now it is recognized the spike protein upon initial exposure. And how long this protection lasts is one of the key pieces of information that we're missing. Now, technology can rapidly adapt if we get mutations from this virus. And there are some common myths. We have heard from people that this vaccine is produced in embryonic stem cells, and this is not true. We also have heard that it's a live vaccine, and again, this is not true. And a lot of people are concerned that it was rushed and not properly assessed for safety. Actually, the studies were quite comprehensive in terms of the safety side of it and the efficacy. The main issue is how long this vaccine is going to last. And that's something that we're still tracking the data to basically understand the duration of the immune response. So there are some references in there if you want to read more about the actual technology of the production of this vaccine. Now, I'm going to go through some steps related to the clinic specifically. Utep sent a questionnaire out, and this was an online questionnaire, and the purpose of the questionnaire is to determine the number of faculty, staff, and students that wanted vaccination and place the individuals in, and this is done by self-reported categories. That way we can prioritize who should get the vaccine first. As part of that questionnaire, there was a consent was obtained for vaccination and also for doing some analysis of the data that we are collecting. And in there, we also included pre-screen using CDC guidelines. And what is the importance of this? Basically, it's ability to plan. And we have been struggling with this in terms of being able to vaccinate everybody that is in the top priority categories because of the response rate was actually very good for faculty and staff, but it was not, it was about 60% for students. So we still have a lot of information lacking. Now, this is from the Texas Health and Human Services website. And I just went yesterday to see if there was any change. And these are the categories of individuals that are going to be vaccinated during this particular phase. So you have 1A and 1B. So within 1A, you have healthcare providers of different types, and also school nurses. 1A also included long-term care facilities, residents of those. And then the 1B is anybody that is 65 or older, or 16 years of age or older, and he has some of the identified chronic diseases that are associated with the more severe COVID-19 disease. So once there is more supplies available, then they will move into other categories. But this is what limits us. We cannot just vaccinate everybody. We have to stick with these categories in order to comply with the state. Now, we do use the CDC checklist for pre-vaccination. Now, for every question that is listed in here, if somebody says yes, we have to be able to explore that a little more. And I'm not going to go through every single one of them because it will take too long. But I do want to emphasize that there are some very important ones. For example, the preference is that if you receive Pfizer vaccine for the first dose, that you should get the second dose within the 21 days that is recommended. You could actually delay after 21 days if necessary, because now there is a larger window of 4 to 6 weeks post first vaccine that still will be effective. But for the most part, we want to keep it as close to 21 days as possible. And then there are some things in here that are critical. For example, individuals that have anaphylactic reactions to other vaccines are probably the ones that we have to be extremely careful. And for the most part, we should not be vaccinating people like that unless they are in a healthcare, comprehensive healthcare facility. So we will not be doing that in our clinic in terms of people that have a previous history of anaphylaxis to another vaccine. What we do know is that it's probably not the vaccine itself, but other components that are used to make that vaccine viable. And there are some other questions in here that are relevant. The last one, pregnancy, is a somewhat of a controversial issue because they are at high risk for developing the disease, the more severe disease. At the same time, there's limited data in terms of what the vaccine may do in terms of a pregnant woman. But what we know is that the data that is available doesn't seem to show any problems at all. So I think the basic recommendation is that that individual talks to the OBGYN, that they are taking care of that patient, but at the same time that they may go ahead and get the vaccine. And if they want it, we will give it to those individuals. Okay, this is a little bit of the layout of how the clinic works. So as you could see, you go through the main entrance, there is a check-in booth, and in here, everything is going to be done using scanners. And you get your ID, and they scan it, and it pops up what information we already have in terms of somebody that is registered to be vaccinated, and the pre-screening of the questionnaire. Once they go, they're checked in, they walk through this area, and then they get to the area where they're going to get vaccines. These are the 5 stations that we have currently in place. So we're vaccinating 5 people at a time. So we do have 2 faculty members and 5 students that are doing the actual vaccination. And it doesn't take more than a few minutes, but at the same time, we need to enforce that the pre-screening questions, if there is anything in there that we need to clarify, but we also use scans in order to track the information of what vaccine was given, when was given, and the arm, because that needs to be documented if it's left or right arm, and so on. Once they finish here, they move into what we call the observation area. And here is where we have to wait for 15 to 30 minutes, depending on the history of that individual. And then after that, they can leave. We do have an emergency medical technician that is stationed here. We have the ability to use a automated external defibrillator if there was a problem. But then the individuals will exit this way. So this is how it's set up now. We're considering expanding and putting additional vaccination stations in this area with additional observation in this area. So that's kind of the flow of the clinic. Transportation of the vaccine is extremely important, and it goes through a very important process of documenting how that is done. And so we track temperatures and movements. Now for the vaccine preparation, so once we move the vaccine from -80 to the refrigerator, we have to wait a period of 3 hours. It is a suspension, so it comes in a small vial and it's frozen, so you have to allow it to thaw. After that, the individuals that are going to prepare the vaccine, they're going to dress with the sterile equipment that includes gown, mask, face shields, and gloves. They prepare the area by cleaning the surface with disinfectant and using sterile technique. They dilute the vaccine using normal saline, 0.9% sodium chloride, and they have to be careful that you don't shake this vial because you can damage the actual vaccine. So you swirl 10 times, and once that is prepared, it has to be used within 6 hours. The amount of the vaccine that is used is, the dose is 30 micrograms and it's in 0.3 mLs. We use a 1 ml syringe, 1 to 1.5 inch needle. Depending on the size of the individual, they may require 1.5 inch needle. And we use a very small gauge, 22 gauge. So the pain is relatively small. We do label the vaccine with the name, dose, lot number, and original expiration. And there is always constant communication from the station to the preparation. And they're very close to each other. So that helps in terms of being able to supply what's needed. Now, this is a picture of one of our students drawing the vaccine. And in there, the reason for using the face shields is primarily to protect them from getting sprayed from the vaccine when you're manipulating the vial and the needle. So that's some people that do not have a lot of experience, that could happen. So the vaccine administration, I have here one of your faculty members from chemistry. So what they do, the students do, is they scan again the ID, and then they clean the surface of the skin, and they go ahead and inject. So that's one of the cards that are given to them, showing that they receive vaccines. This is a CDC card. In this case, I think you may recognize who that individual is. After the vaccination, he came to me and asked me to touch hands in a distant kind of way. The observation area, again, is 15 to 30 minutes. 15 is for everybody. 30 minutes is for somebody that has an anaphylactic reaction to another medication, not another vaccine. And in this area, there is a little bit of education in terms of what to expect from the after the vaccination. We do ask participants to sign up with the V-Safe, which is an app, a phone app that was developed by the CDC and is to track and remind people in terms of how their next vaccination is going to be, and also they send information about if they're having any kind of side effects or problems. So this is a national type of database. We are doing 2 research projects. One of them is a survey on perceptions and beliefs related to vaccination. So that's actually done there during the observation time. They can participate on completing the questionnaire. We do have another project, another research project that is more about effectiveness and safety long-term of the vaccine. So that is done via email. Now, the EMT is prepared to give emergency medications, and this is a short list of some of what they have in their backpack. The most important one is epinephrine, because somebody has an anaphylactic reaction. That is the drug of choice to treat it. But they do have other medications, and we do have a few patients that require Benadryl because they were developing hives. Vasovagal reactions are also from people that are a little anxious and they start hyperventilating. We have some cases related to that. But basically, the EMT is there to manage those patients. Now, reporting, there's a lot of reporting that has to be done related to the vaccination clinic. The MTRAC, I already mentioned, that is for the state tracking of vaccine administration. There is also the Texas Division of Emergency Management. This report actually provides the state, the numbers of vaccines that we were able to administer, so they can reallocate more vaccines for future use based on how many we are actually administering. Now, there is a possibility that somebody has a reaction, and there is a system that we need to report to. It's called the Vaccine Adverse Events Reporting System. This is a system that is developed by CDC and FDA, and there are two types of reactions that they are tracked: adverse events and what's considered to be serious adverse events. The serious adverse events are more like they require treatment or hospital, or, of course, if somebody dies from the administration, that will be the ultimate most serious side effect. But we have not had a single one of those serious adverse events so far. And so far we administered a total of about 4,500 vaccines. We're in the process of continuing vaccination this week. Last week, we did not get vaccine because of the weather. So that puts us behind in terms of some of the participants that were going to receive their second doses. We do have a very extensive handbook of operating procedures that covers 50 pages of detail for how we're doing things and forms. And of course, a lot of what is in there is training. Each one of the stations that you saw have a specific training that needs to take place. Now, some of the training has to do with more of the operations of the registration to, observation and so on. And other training is more clinical, like the actual administration of the vaccine, the screening, using the screening of the CDC, and how to answer those questions that may come up. There is also the training in sterile technique to prepare the vaccine. So, and also the storage of the vaccine, because it's a very delicate vaccine. Now, as part of that, we already have one needle stick. So I want to emphasize that we do have a procedure what to do with needle sticks. We use retractable needles for most of the administration. And those basically, once you inject and you push all the way, the needle retracts back into the syringe, which is a different technology in terms of preventing needle sticks. But we still have one case of a needle stick, and that needed to be addressed because of the risk related to needle stick that involve potential blood contamination. The communications component of this is very comprehensive. I emphasize primarily the clinic, but there is a program that requires a lot of information that goes through either emails or also the batching of the data, the scanning, the process of developing all these apps and abilities. So there is a lot that goes into communications. As part of that handbook of operating procedures, we do have instructions for faculty, instructions for students, and it goes on. I want to emphasize the observation. We have to talk to some of the students that volunteer for the observation area, because part of it is, educating, but part of it is actually observing, because we have two individuals that fainted, and it was more related to anxiety than anything else. But that potentially can be harmful if somebody falls and hit themselves. So we have to follow a few patients related to some reactions like that. But overall, there's only 2 people that received treatment with Benadryl, and it was for hives or itching. And so far, we have a good experience doing this. Utep is considering expanding, but it will take hiring individuals to run a larger clinic. So these are some of the issues that we're still working with. Now, because we don't know enough about the duration of the immunity of this vaccine, data is coming out, and new information is coming out, and studies are being documented, especially in the media, and we cannot make changes until things become official. For example, there is some studies that have shown that maybe a single dose of the Pfizer vaccine is sufficient, and maybe we should be using more single doses instead of having to use second doses for the same individual. Well, this cannot become official until we get guidance from the state. And usually the state follows guidance from the CDC. So sometimes the media puts information out that can create a little bit of confusion, because studies may show a different result. And the same thing with the variants of the COVID, or this particular coronavirus. We still don't know how effective our current vaccines are, but there is data, conflicting data. So until it becomes official, we just have to be careful with the interpretation of what's in the media. So this is one of the first days that we had at the vaccination clinic. One of our students are in here in blue, on the dark, and then the faculty is on a white coat. So that's one way how do we differentiate the faculty from the students, is when you see the white coats, that's part of the faculty team. So with that, I'll stop and see if there's any questions.
Chair, can I answer the first question? Paulina?
Yeah, go ahead.
Yeah, actually, I feel actually we as a Utep faculty or student, we are very lucky to have this vaccination program, thanks to Dr. Rivera, as well as we have the campus testing. So these two things is almost like a significant benefit for the Utep student and faculty. And during yesterday's our discussion Q&A, actually there is a question, because this vaccine actually based on research is not preventing infection. It's possibly just preventing severe disease. So Dr. Kirken is here. So we have a question, is that possible to link the vaccination program data with the testing? So to trace all those people has been vaccinated, whether they still got the infection, because there's some studies show that even you're vaccinated, you still can get the virus tested positive by the PCR test. I'm just wondering whether some of this data can be passed to the testing program to combine these two benefits in the campus.
I don't know if Dr. Kirking is going to comment on this, but I did want to comment that the second research project that we have does ask if individuals have tested positive after receiving the vaccine. So we want to track that information. but it's self-reported. It's not actually documented that we get that information, that the test was positive.
Yeah, no. Well, first of all, I mean, just thanks, Paulina, for organizing this seminar. I think it's fantastic and timely. And thanks, Jose, I think, for, just amazing leadership here on this topic. And of course, with all your other duties, I mean, this is really spectacular, as I think River said. But yeah, no, that's a really good point, River. And we are trying to do that. We are trying to match that up. The challenges are the IRB issues. This is a big challenge. And trying to get release for those to participate. Of course, as everyone knows, both testing and vaccination is purely volunteer. And so that is the bigger challenge for us. But I think some of the survey work that Dean Rivera is doing hopefully can shed additional light into that subject area.
And we did have one case of an individual that received the first vaccine and a week later tested positive. So he probably did not have the full immune response from the first dose, but the recommendation is that once they are out of quarantine, to get the second dose. So that was the one case that I know. But I'm sure that there could be others and we don't know.
Maybe I'll ask a quick question, Dean Rivera. I'm just trying to think about the timeline here for Utep. I'm not great at math, but let's say we have about 30,000 Utep faculty, staff, and students, and let's imagine that no one gets a vaccine outside of Utep. Let's just say it's a pure 30,000 number.
Yeah.
And if everyone needs 2, that's 60,000 vaccinations, and you divide that by 1,000 per week or so that you're coming in, so that leaves you with about 60 weeks. You divide that by 4, that's about 15 months, I guess. What's your thought about,
yeah,
can we accelerate that timeline or take more doses, or what do you think, so that we can?
Yeah. Let me comment on that, because I have been thinking about this for a long time. The first thing is we already vaccinated, based on the reports that we received, everyone at Utep that is considered to be in the categories of high risk. So that on the one side, yes, we still have, right now we're at, 4,500 doses given, including the first, second dose group. So yeah, it will take a long time for us to vaccinate everybody, but we have been able to at least take care of the highest risks people at Utep, based on the self-documentation and desire to get the vaccine. The number of people that say, no, I don't want to get the vaccine, was relatively small. But there is a lot of people that did not respond to the surveys. Now, this coming week, we are going to vaccinate more cases. I'm happy to inform everybody that nursing, the School of Nursing, has finally joined us in terms of, actually having faculty and students participate with the vaccination program. Right now, it was the School of Pharmacy and the College of Health Sciences, the Clinical Laboratory Science and Public Health as the volunteers for the clinical side of this clinic. And finally, we got the School of Nursing to get on board. So this coming week, we are planning to vaccinate more than 3,000 people. Well, that includes second doses, but it's close to 3,000 people, let's say. So we are increasing somewhat the capacity. We do know that some people are getting vaccinations outside Utep, so that decreases a little bit in terms of the total number. And Utep is negotiating to try to become a hub. A hub is a place where you can vaccinate 1,000 people a day for at least 5 days a week. But that requires additional personnel. But I mean, if you want to say, well, at least we are taking care of the highest risk people, that was accomplished already, at least from the point of view of the people that submitted the information through the survey. And it was self-reported. And there are conditions that put you at high risk, besides being older, like cancer, in pregnancy, there are sickle cell disease. There are several different conditions that put you at high risk. And so if people did not respond, then that's, you know, they're missing the ability to get the vaccine.
Thank you.
Okay.
I have another question. Is that I heard that the Pfizer put a little bit more actually in the vial. So it's possible we got the additional a little bit based on the practice?
Yeah, actually, for the last, since the beginning of Pfizer started sending vaccines, the total number of doses that they were official was 975. And that was based on 195 vials, being able to get 5 doses per vial. So very quickly, it was recognized that you could get 6 doses per vial. This is not uncommon. This is called overfill, which is something that is done commonly for medications, injectable medications, where you have a little more volume, because you know, there are issues related to, depending on the needle size that you use and things like that, that you can have, you know, when you draw them out, you need that overfill. But on the other hand, now it's official. As of this coming week, the total number of doses now is formally based on 6 doses per vial. So there is, it went from 975 to about 1150. But this coming week is when it's actually official. This was approved by the FDA already, and now, like I say, as the next batch that comes, it is a larger number of doses in those 195 vials.
All right, Paulina, I've got a couple of questions, if I may.
Yes, of course, go ahead. And then we'll go into the chat.
Absolutely. First of all, thank you very much for organizing this, and River as well for leading the chapter. And I thank both the deans for being here today. The first question is for Dr. Rivera, and then the second question is for Dr. Kirkin. And I'm going to actually ask both the questions straight away. Dr. Rivera, there is a thought that those folks who actually got COVID, whether they need the vaccine simply because beyond the spike protein, they've been exposed to everything that comes with COVID, right? All the insides of the virus, and your antibody regimen will be much more robust than just the antibody towards the spike protein. So do those folks who already have COVID or have had COVID, do they need the vaccine? And then the second question for Dr. Kirkin is on the testing. When somebody tests positive, what is the sequence of events that take place? Do you all inform the EHS, et cetera, et cetera? So Dr. Rivera first, please.
Okay, well, the current recommendation is that we do not know the natural immunity of the disease, how good that is. So for the time being, it is recommended that they still get the vaccine. Now, they may not be at the highest risk, because the expectation is that they do have some immunity, but we just don't know how good that natural immunity is. So that's one issue. So in general, especially if you have other conditions that puts you at the highest risk for more severe disease, is to go ahead and get the vaccine, even if you have the disease. But you may have some protections already from natural immunity.
Dr. Kirkin.
Yes, no, thanks, Mahesh. So there's a number of things that go into play. So I think everybody knows that as far as the testing is concerned, everyone's linked to your Utep ID. And then when you get when you go to register for the test, you get assigned a number, a barcode basically, that keeps the anonymity of that entire process. And it also, the way we built the testing centers, that barcode actually corresponds to a well in that 96 well plate. So it's maintained all the way through from when we take the sample, isolate the RNA in that sample, switch to another well for the PCR. And then when we obtain the results from that test, we actually put it through, it's an approved interpretive software platform that determines positive and negative, or if it's an invalid. And then that goes into another pipeline at Utep where IT controls remerging identity with that sample. And at that point, essentially what happens is a couple of things. So EH&S is alerted. There's a COVID committee that is alerted about who tested positive, along with who tested negative. As you know, those results are disseminated to the individual. But at the same time, the positives are also reported to the city through county public health so that they can do contact tracing. But on the Utep campus, EH&S and various people are involved in contact tracing here on the campus. But a number of things go into place. Everyone's identity is still maintained so that no one's name is provided, but it initiates a number of things. So first and foremost, trying to figure out if that individual's been on campus or not, which will trigger another level. So a lot of times, whoever it is, student, faculty, staff, their access to buildings is pulled almost immediately so that they're not allowed to re-engage for that quarantine period. And at the same time, usually it triggers a clean response. So there's a dedicated team that goes in and cleans those areas where that individual was. And deans and chairs are often alerted that there was someone who had tested positive, and it may be within a particular area, but those are the series of events that happens once someone is identified as a positive case.
Got it. Thank you.
I just want to clarify a little bit. So Mahesh, this event is really purely initiated by students. I'm very proud of them, okay? So, and I forgot to mention, Christine actually also did a lot of work. Christine first contacted Dr. Kirken, and Kirken referred us to Dr. Rivera, and then the whole thing started. So, and Christine met with Dr. Rivera. So those student officers, I'm very proud of you guys, okay? Those are those with ASBMB background. Those are all our student officers. So they did organize all this. I did nothing, okay?
I have to agree with that. So thanks to the students, it's a pleasure to be here. And I hope that this helped in terms of clarifying some of what we're doing and how it's being done. We're as careful as possible in terms of protecting the vaccine and protecting participants and protecting the volunteers, which includes faculty and staff and students.
So
Can I
thank
ask
you.
a question?
Oh, sorry. I just want to say thank you for acknowledging the hard work of the officers. I agree. Christine did most of the contacting with Dr. Rivera and Dr. Kirken and organizing the event. Is anyone gonna ask a question? Because if not, I'm gonna go to the chat questions.
Yes, can I ask a quick question? First, I'd like to thank Jose Rivera. Jose, this is Jorge Ardea. Thank you for the great job. And as you know, I work in the area of nanotechnology. And one of the questions I have, I know the process where how the vaccines were developed. You know, the Pfizer vaccine is a 75 nanometer lipid and encapsulated vaccine. Supposedly every shot contains 30 billion nanoparticles in every shot that you guys do. So my question is about the safety. How do you make sure that the vaccine is working? You know, the freezing process of -80, you know, could have been something could have been wrong. So before you apply the vaccine, how do you guys make sure that the vaccine is good?
Well, we're following very strict guidelines and procedures that are outlined by the Pfizer themselves in terms of how we do the transportation of this vaccine and the documentation that every step that was, once you move the vaccine from the beginning, what we started doing was taking pictures of when we receive it, when we open the container and we move it to the -80, the picture documents the time that it took to move it. And we have that 3-minute window. And the same thing when we bring it from the -80 to the fridge, where we are tracking. We have an inventory control form, which I did not share here, but if you see it, it shows every single step of the movement, and that's cold control. As we move it from one place to another, when didn't we move it? What was the temperature of the freezer or refrigerator, and how much time we are allowing for each one of the steps. So this is all documented. I have a pretty amazing form that was developed for this purpose because it documents every step.
Right. So, as you know, I'm a chemist, and I teach my students. Actually, I'm teaching a class on nanotechnology now. We cover quality assurance. So, in chemistry, when you get a sample, you do the quality control, making sure that whatever is supposed to be is there. But I can see from your answers that there is no chemical or biochemical tests, right? And actually, Pfizer just announced this morning that the temperature could be not needed. I don't know if you saw the news.
Yes.
That the -80 may not be needed. So.
Yeah, and that's.
Yeah.
I mean, these are. There's a lot of changes happening with the vaccinations, and how good are they? How long are they? Do they spread the disease? Do you contain spreading the disease? There are evidence out there, but until it becomes official, we cannot change it. So we continue using what's established as the official way of managing this process. So you're right about some of the evidence that is coming out, but we have to stick with what's official. Because we could be making a decision on a premature basis. So we will have to wait until it becomes official for us to make changes in our cold chain control process.
Yeah, and just to kind of jump in there, Jorge, every drug goes through this, usually has a much longer timeline for production, right? So there's usually years and years of preparation, and of course, monitoring and looking at drug stability through that period of time. And all of those factors, heat, temperature, humidity, all of those things are being assessed. And of course, this is not the case, right? So remember, this is not really an FDA-approved drug, right? This is an emergency use authorized drug. So most likely, all of these things are being very much calculated and monitored and measured, especially on the stability side, and especially with those vesicles and nanoparticles. So I think you'll see more and more data coming out, and that will probably change how this system is going to be applied in the future. But what I think is really exciting for you is that these nanoparticles, this is probably going to be the next generation, the next wave of drug delivery in many cases. So I think it's really important that we monitor this, especially thinking about gene therapies and delivering other types of drugs. These phospholipid carbohydrate-based vehicles are going to generate a ton of data and could really jumpstart a program as far as drug delivery and new mechanisms.
So this is a follow-up question for either of the deans, and if it's sensitive, please punt. Given the fact that the tracking is down to the third decimal, how come the shipments were lost the first time around by the city or whatever? Remember, there was a cancellation.
I'm not familiar with. We didn't lose.
No, not
we
you.
have.
I was just curious what happened there if you're new. And again, if it's sensitive, we don't have to discuss this.
Yeah, I'm not sure. Basically, we are treating the delivery of this vaccine like if we're receiving controlled substances. We have security waiting there because we understand that there may be a price that somebody is willing to pay for getting 975 doses or 1150 as the new regulation. So we're treating this at very carefully, and we have security for receiving. And they actually go with us all the way to the freezer. Once we put them in there and we have, lock the freezer, then they leave. So that's how sensitive we are about receiving and storing that vaccine.
And maybe just to chime in there, Mahesh, it's a logistical issue, right? More than anything else. I think this is the real challenge to overcome. I think everybody on the administration side with the delivery of the vaccines, as far as to the individual, is very efficient. I can tell you that I also sit on the city COVID-19 task force, and we often look at how El Paso as a whole is doing in delivering and executing the vaccinations. And I can tell you, based on the real numbers, El Paso is the most efficient city in the state of Texas right now in doing that. So I think, as Dean Rivera said, as soon as those vaccines come through the door, it's very well executed, that delivery system, and especially here. So, but how it gets here, that's a little bit beyond our control.
No, I was not doubting the 79968. I was just inquiring what happened the first time around. That's all. If you all knew what the city or what had happened.
Maybe last question, if I may, is in our virology group, we are actually has some kind of observation, might not be true at all. It's some of us discuss if you get vaccinated at different arms, you have less side effect. And we, at least in our small 100 virologist group, seems to be true. I'm wondering whether Rivera, you have seen like this kind of observation. I think CDC should have answered immediately because they did a survey of us, what kind of side effect it is, and you guys actually register which arm we got the shot, right?
Yep. It is a requirement that we document which arm the administration of the vaccine is given. And actually, the second time around, they can choose one or the other. We cannot tell. It has to be the opposite or it has to be the same. It's up to the individual, but we do document which arm. Okay.
So if we can go to the chat questions just very briefly. It says, at the current rate, when will vaccinations be available for those under 65 with no underlying health conditions?
Well, if you go to the website of the Texas Health and Human Services, they're talking about late spring, there will be broader distribution. The Johnson & Johnson vaccine now is under review for another emergency approval use. And then Mrna should be in the pipeline. So you have other companies that they are producing vaccines. They are different. This, the upcoming vaccines are different in terms of the technology and also the requirements for storage. So that we'll have to see. And of course, Johnson & Johnson is a single dose. Again, if you go to the website of the Texas Human and Health Services, they are talking about late spring where there will be more dissemination of the vaccine for other populations. But right now, they're still holding this to those two categories, 1A, 1B.
Okay, thank you, Dr. Rivera. And the next question is...
Can I interrupt you because that was my question?
Yes, go ahead.
Because that answer does not make sense to me at Utep right now. If you say you've already, vaccinated everybody in 1A and 1B, and you're just vaccinating people at Utep, who are you going to be vaccinating the rest of February and March?
Okay, so right now, primarily what we have is second doses. But starting the first week in March, one of the things that Utep is doing is reaching out to other partners in the community that have 1A, 1B. And that's how it's been approached, because I think the president is very sensitive that if we stop vaccination, we may not be in the priority list of receiving additional vaccinations as this opens up the gates for other populations to be vaccinated.
Okay, so everybody in El Paso who's in 1A and 1B is going to be vaccinated first before anybody who's not in that group at Utep is vaccinated. Okay.
Yeah.
Thank you.
The next question is if the vaccination program will actually reach family members of the Utep community.
The expectation is yes. That is part of what they are planning to do. And how the logistics, that still needs to be worked out. You know, I function more on the clinical side. Some of that is more on the operation side of the program. But I know that that's an expectation. And I don't know how soon we're going to do this. And some of the logistics have to be for a UTEP employee or student. It's easy because you have a scan that has all your information. When you bring people from the community, then there is a more labor-intensive process of, you know, not only bringing that person for vaccination, but also documenting that that individual is in the 1A, 1B group. So there are more logistics, but the expectation is to bring some family members.
And the next question is, if you know, what are the biggest manufacturing challenges for the Pfizer vaccine?
You know, I'm not an expert on that area. I don't know if Dr. Kurt King or anybody else. My understanding is that making the messenger RNA is not that difficult. Creating the delivery system, that's where the challenges are. But I'm not an expert in that area.
Yeah, making the liposome may be hard, and but I heard today at the news that Biden actually just visited the Pfizer site. They are going to boost up the production line. That's what I learned this morning.
River, can I comment, because I know about what is the big challenge there. So the big challenge is the test to make sure that the nanoparticle is 75 nanometer. And so they developed this DLS technique to determine the nanosera potential, that the distribution of the nanolipids is almost the same as the 75 nanometer. And the reason I know is because I studied that, I work in that area. And the other issue is my daughter works for AstraZeneca. Jose, you remember Jessica, right?
Yes,
She's with AstraZeneca. So AstraZeneca is developing, I mean, the vaccine from AstraZeneca is a different vaccine. It's a viral that is the size of 90 to 100 nanometers. And they have the same issue with the distribution of the size. Basically making the particle at that size.
Thank you for answering the question. And the last one is from Dr. Beckbar. It says, some time ago, Dr. Rivera said he did not understand the requirement for negative 80 degrees and the difference between the two vaccines in distribution. So far, Dr. Rivera, any updates in understanding? Does this have to do with the liposomal nature? And Dr. Beckbar also asked if there is any information on how many or how much mRNA molecules per liposome particle.
No, those I don't know. But at the beginning, I don't understand why Pfizer need -80, because both are liposome based. So I really don't understand. I have a suspicion, but it's not approved. Okay, they might use some LDLP on that lipid, because not the liposome, not every liposome can just fuse with the cell, get the mRNA in. There must be some trick. So they must be using some trick. Maybe they put some LDLP, which is some protein actually deliver fatty acid to our different organ tissue. When you have protein, then I can understand. Sometimes in order to make sure that the protein perform right, then you need to make a -80. That's my suspicion. There's no proof of that at all. So, and we still don't know what kind of technique. I think they don't publish that. It's a proprietary kind of feature of that liposome. So,
I just want to make one statement that most vaccines require refrigeration or freezing, but not -80, -20. So this is unusual, and I don't know either, why they have to do it using -80.
And the last question comes from one of our members, and he asks, what kind of cells are the ones processing the mRNA, translating into proteins to present to the lymphocytes? Any type of cell or just antigen presenting cells?
wow. Rob, can you answer that? That's kind of very biochemistry question. So I think we don't understand which cell those liposome deliver the mRNA. Very possible some just the arm, those muscle cells, normal cell. Then they got the information, which is like our first innate immune system. Then maybe the T cell, the macrophage cell arrive and kill those cells, get those protein out of their surface. That's my guess. So,
Yeah.
Rob?
And I'm not a true immunologist anyway, so my background is more in chemistry. But anyway, no, because remember, that is an injection into the muscle, right? So most likely those cells, it's our cells that are translating that mRNA into protein, right? That is going to be expressed on the surface of our cells. So it has the proper folding, post-translational modification, et cetera, et cetera. So most likely it's our immune cells, and especially antigen-presenting cells, that are recognizing that as a foreign antigen, right? So they're gobbling that up and then presenting those on their cells and activating T cells, and then that consequent B cell response to generate the antibody. So, so that is the reason, but it's not a delivery just to purely activate immune cells, right? It's part of that entire process, but clearly that's why your arm gets sore, right? Because that's an immune response that's going on. A lot of inflammation and all of that is critical for the immune response that you're going to develop for long-term protection.
Yeah, I do want to add a little bit. End of last year, I do see a TEN image when that liposome was delivered to a cell culture. They cut the cell into a section. Look around the TEN. I was so impressed that the whole cell is covered by that spike protein. Yeah. So the translation is really, really good. So.
Mm-hmm.
Well, I just want to add, in the end, Luca, the whole thing needs to be transferred to the lungs, because that's where you get the first shot of COVID, right, to the respiratory system. And in this context, it's very important. And if you were there in River's talk, actually it wasn't a talk, it was a discussion by 4 faculty and the dean. The actual spike protein that is being manufactured via the mRNA is a mutant, which has 2 prolines. And that prevents the spike protein from doing a Swiss knife-like procedure, right, and entering the cell. Now, if the spike protein actually did that, the immune system would not have had a chance, given the time that it takes to be ready, right? And the spike protein would have actually entered. So you have actually prevented via these 2 prolines, the Swiss knife opening of the spike arm, and you give the immune system enough time to generate antibodies and then percolate through the lungs, etc. There might be a Nobel Prize there. The UT Austin people, they were speaking about it in the first seminar of chemistry. It was very interesting.
As we conclude our meeting, I want to thank Dr. Rivera for giving this very interesting talk and answering all the questions that we had. The Dean of the College of Science, Dr. Kirkin, thank you so much for being here. And to everyone that in general was present here. Before we go, can I ask you, some of you, to turn on your camera so that we can take a picture? We usually upload the pictures to our social media. So yeah.
I want to appear in Dr. Rivera's next presentation. Make sure I'm there.
Okay. I see that some of you are turning your camera on. Thank you.
Jacqueline,
Okay.
Osma, can you turn on your Domingos, Daniela?
You need to take two
Lucia?
screenshots, the next screen as well.
Yes,
Yeah,
because we
there's
have two screens. I'm
additional.
going to be taking two
Yeah.
pictures. Okay, so I'll go ahead and take the first one. If you want to do a picks up, that's totally fine. Okay, 1, 2, 3. Okay, now I'm going to go to the next screen. Let me just wait for it to upload. Okay, again, 1, 2, 3. Okay, awesome. Thank you to everyone that attended. This was really awesome. I will be uploading this recording to our social media. I'll make sure to share it. And please, ASBMB members, make sure to stay. Everyone else, thank you for being here. Have a lovely day.
Bye-bye.
Thank you. Thank you all.
Bye-bye.
Thank you all.